Good afternoon, everybody. First, let me start off by thanking Subith and Michael and Jennifer for putting this all together. It's been a really great series so far. My name's Lorraine Leon, and I'm in the Materials Science and Engineering Department. I'm an assistant professor, and I've been at UCF for about 3 years now, a little bit over, since 2017. So today I'm going to introduce myself and discuss my lab's research interests at the interface of biomaterials and polymer science. Okay, so first a little bit about myself. I'm a chemical engineer by training, and so I have a bachelor's degree from the University of Florida and a doctorate from the City College of New York, which is part of the City University of New York system. After my PhD, I moved to Chicago and worked at the Institute for Molecular Engineering, which is part of both Argonne National Lab and the University of Chicago. At UCF, I'm at MS in Material Science, as I mentioned before, and I'm also affiliated faculty with the Nanoscience and Technology Center. On the education side of things, I teach both undergraduate and graduate courses in polymer science, as well as the Intro to Material Science class called Structures and Properties of Materials. I was also part of the committee responsible for developing the new undergraduate program in material science, which launched in fall of 2019. On the research side of things, I'm interested in peptide-based materials, self-assembly, polyelectrolyte complexes, biotemplating and biomineralization, small angle scattering, both with X-rays and neutrons, as well as general polymer characterization techniques, most of which you'll hear about in the next couple of minutes. So the overarching goal of my laboratory is to mimic the flexibility, adaptability, and overall dynamic nature of natural biomaterials that are made from proteins. Proteins play fundamental roles in work performed by cells and the structure and function of tissues and organs. The way natural biomaterials achieve this dynamic nature is through a series of non-covalent molecular interactions that are carefully tuned. Our approach involves using peptides to create dynamic materials. We do this because peptides can be designed to incorporate a plethora of molecular interactions due to the functionality of the 20 natural amino acids and even more from unnatural amino acids that are being designed every day. For example, as shown below, we have electrostatic interactions from charged amino acids, hydrogen bonding from the peptide backbone, and a host of other molecular interactions that we can introduce. We choose to design peptides because they're easier to design and synthesize than full proteins, but they also allow sequence specificity, unlike other soft materials like polymers. Now, obviously, this strategy can be used to allow to design a variety of different materials. But today I'm going to focus on this one example that I've been excited about, and that is membraneless organelles. Membraneless organelles are highly concentrated micron-sized droplets that form and dissipate within cells. They form by liquid-liquid phase separation, and they lack a lipid bilayer, as their name suggests. They're involved in a variety of functions, some of which are being discovered every day, like stress modulation and signal transduction, but they're also implicated in disease. The concentrated droplets are made of proteins and nucleic acids, where intrinsically disordered regions in the protein appear to be driving the droplet formation, but the exact molecular origins of the driving forces are poorly understood. Here, I show you a membraneless organelle called the Nucleolus that forms within the cell nucleus. And as you can see in this video, it forms various layers of concentrated liquid phases. The Nucleolus is responsible for the assembly of the ribosome, which is done from the inside out, where each layer has its own function. Basically, it's like a dynamic biological reactor. In our work, we use peptides to understand both the driving forces of this phase behavior, but also how we can mimic these transient dynamic structures. Okay, so with that background in mind, the first project I'm going to discuss involves making these concentrated liquid droplets called complex coacervates very similar to the membraneless organelles I just discussed. These are formed by mixing oppositely charged polymers, which sometimes create these droplets, as you can see here, and sometimes they create these glassy aggregates, as you can also see in the bottom. Phase behavior is also poorly understood. The main driving force for these droplets is the release of counterions that occurs when the oppositely charged polymers come together. With peptides, we have shown that you can tune between these phases by introducing hydrogen bonding. Our current objectives are to fundamentally understand the effect of increasing hydrophobicity on these droplets, which we can easily accomplish by designing different peptide sequences. Since these droplets form by mixing oppositely charged polymers, it is easy to incorporate things like negatively charged RNA or DNA for applications like gene delivery. But incorporating hydrophobic drugs and dyes has been less explored, which is another one of our objectives. So far, we have designed and synthesized 12 new peptides that form 6 different coacervate phases. These phases have shown enhanced stability with enhanced hydrophobicity. We have also shown that with enhanced hydrophobicity, you can form more droplets with increasing temperature, which is a behavior that is opposite of non-hydrophobic complex coacervates. Increased hydrophobicity has allowed increased encapsulation of hydrophobic dyes and drugs. Some examples are methylene blue, as you can see here, doxorubicin and camptothecin, which are anti-cancer drugs, and dye, which is a commonly used imaging dye. We have also shown that you can control the phase behavior by manipulating the chirality of the amino acids used in our peptide designs. And this is important because we want to obtain this droplet phase that can be dynamic. We have collaborated with Sharon Laverty at the College of Staten Island on molecular simulations of these different peptides, which is helping us explore some different sequences without having to first synthesize synthesize them. In the future, we want to do some rheology on these different phases with and without encapsulants, specifically microrheology, which gives us the advantage of not having to use too much material. Due to the fundamental nature of this project, we're exploring the NSF as a funding source. Somewhat related to the previous project, the next project uses peptides we have designed with different levels of hydrophobicity to recreate the structure of the nucleolus that I told you about previously. Our overall objectives are to determine the material properties correlated to these multiple layered formations, which in the case of the nucleolus is driven by differences in surface tension. In addition, we would like to develop the capability to encapsulate different materials in different phases, where these molecules can be proteins like enzymes or hydrophobic drugs. So far, as shown above over here, we've seen how you can encapsulate different materials in different phases, but also if you mix a more hydrophilic coacervate with a more hydrophobic coacervate, you can form these multi-layered structures. And here you can see using confocal microscopy that the hydrophobic coacervate is in the interior. We have also shown encapsulation of different dyes, both tethered and non-tethered to the polyelectrolytes. In the future, we're looking to encapsulate enzymes and use these different phases for different functions, mimicking the function of the nucleolus, but for other applications, such as the synthesis of biofuels and pharmaceuticals using enzymes. To verify enzyme encapsulation, we're going to use Raman spectroscopy and have enlisted the help of Alphonse Schultz in physics. We are also measuring the surface tension of the different phases using the Cecil drop method and confocal microscopy, but are exploiting things like colloidal probe AFM to do this as well. We are also exploring funding through NSF for this project. All right, the next project I want to talk about describes using the same polyelectrolyte complex, but now they're formed on the nanoscale. This is done by conjugating a non-ionic water-soluble polymer to either the cation or the anion, or both. When you do that, you can stabilize the phase separation on the nanoscale, and depending on the ratio of the non-ionic block to the charge block, you can design micelles of varying shape, either spherical or worm-like, as shown here. We have developed both solid and liquid core micelles and also shown the encapsulation of nucleic acids and dyes for applications in drug delivery, which have shown some promising results both in vitro and in vivo for the treatment of atherosclerosis. Currently, we're exploring the effect of having a thermoresponsive corona, which has a hydrophobic to hydrophilic transition, and how this will affect morphology and stability of the micelles. We're also looking to develop the ability to use temperature as a trigger to localize and slowly deliver cargo, which could be done using things like high-intensity focused ultrasound. So far, we have carefully characterized both worm-like and spherical micelles using a host of scattering techniques, such as small-angle X-ray scattering, as shown on the top right. Above the temperature transition, we show that the worm-like micelles lose their structure, but begin to aggregate, while the spherical micelles retain their structure, but also aggregate. We have shown that with solid core micelles, these assemblies do not exchange with their environment, and thus can slowly deliver cargo in specified locations. For this project, we've collaborated with Xiaobing Zhuo, who's a beamline scientist at the APS at Argonne National Lab, where my group has been roughly once per year since starting here at UCF. In the future, we're going to be doing some neutron scattering on these assemblies, and we were recently awarded beam time at Oak Ridge National Lab. We're also interested in exploring the intracellular trafficking of these materials and would welcome any collaboration in this area. For funding, we've explored things like the Oak Ridge Associated Universities funding mechanism, Johnson & Johnson, National Science Foundation, and in the future, looking at things like NIH. Lastly, I'd like to talk about how to use biomolecules to direct the growth of inorganic material. As you know, nature controls inorganic growth of bones and shells with incredible precision using proteins. Here I show an example of magnetotactic bacterial that synthesize iron oxide that bacteria use to arrange themselves in a magnetic field of the Earth. Our goals are to study the effect of peptide templates on the morphology and crystallinity of these inorganic materials, and also to develop the ability to organize these into larger structures beyond the nanoscale the way nature does. So far, we have used simple templates to grow iron oxide into needle-like and spherical structures using just polyglutamic acid and polyelectrolyte complex micelles that I described previously. We have also learned ways to characterize these hybrid materials, and I've been invited to serve as a guest speaker to strategize on larger scale grants at Georgia Tech. We have partnered with Blair Bretman at Georgia Tech, looking at peptides to control polymorphs of pharmaceuticals, which is a future direction we're looking at. We've also looked at ways of performing in situ, we're looking at ways of performing in situ characterization, so we can see how these inorganic phases develop. For funding, we have been exploring a specific call that was in DARPA, Air Force, and also NSF. With that, I'd like to thank my research group, in particular Sarah Tabande and Sasha Shaw, who are my first two graduate students and were very instrumental in getting the lab set up. Both of them will be graduating next year. I would also like to thank my collaborators and all of you for your attention. I'd happily answer any questions if they arise. Thank you.
All right, thanks, Lauren.